Fungi for Function is a 3-year citizen science fungi research project, and we're currently analysing the data from our very first pilot year that we've just had, so yeah, it's quite an exciting time at the moment. So yeah, I'm Sophie Green, I'm the project coordinator for this project, and we've also got Dr. Jasmine Packer here in the audience as well, who's the chief investigator for this project. So the project is funded by the Due Citizen Science Large Grants Program, which we're very grateful for. The strength of this project is the range of complementary project partners who are actively contributing. So the project was co-designed with Dr. Wendy Stubbs and Nicola Barnes from the Murraylands and Riverland Landscape Board, mycologist Dr. Teresa Lebel from the State Herbarium and Dr. Sapphire McMullen Fisher from Fungi Ecology. We've also got ecological modeller Dr. Thomas Prowse from the University of Adelaide involved, and it was also co-designed by citizen scientists such as myself. It builds on Find Our Fungi, which is a foundational project by the Murraylands and Riverland Landscape Board and a range of other partners. And Alice, who spoke earlier, was very involved in that project. So as a national fungi citizen science organisation, FungiMap is coordinating the project, and I work for FungiMap. And we're also working with Culture to Country Consulting as First Nations advisors, with our data partner, the Atlas of Living Australia, and it's great to have Erin here today from the Atlas of Living Australia, and with a range of site partners. So we've got Trees for Life, the National Parks and Wildlife Service, Second Nature Conservancy, Hills Biodiversity, and the Wollonga Environment Centre. And we've also had Jess from Wild Webcat helping out at our surveys as well. And I think this teamwork is what has made the project go as smoothly as it has in this first year. So very little research has been undertaken in Australia about the role of native fungi in our ecosystems. So this project is looking at whether recording the richness and ratio of target species within key fungi functional groups can indicate ecosystem health and resilience in our native bushland. And the 4 fungi functional groups that we're focusing on are the plant partners, or the mycorrhizal fungi, the recyclers, or also known as the decomposers or the saprotrophs, plant parasites, and also lichens. And we're predicting that a balanced proportion of fungi species across these functional groups will correlate with healthy bushland. As there's so little knowledge about Australian fungi and their conservation status, a byproduct of the project will be new data on the distribution of distinctive target species and their abundance in different rainfall and habitat conditions. So to answer our research question, we're trialling systematic fungi surveys with citizen scientists at 10 sites throughout the Mount Lofty Ranges, which are across a rainfall gradient. And we trialled 3 sites this year, and we're building up to 10 sites in year 3. So the systematic design elements are the same in every survey, and these include site criteria such as the vegetation community and the aspect, and the search effort also stays the same, so the area that we're surveying and the amount of time people are spending surveying. And the test design elements vary in every survey, including rainfall and habitat condition. So we're looking at good condition and poor condition due to a range of disturbances, so that we can test the fungi found in each. And in the survey, citizen scientists are specifically looking for these 25 target species across the functional groups. And the reason for choosing target species is that they are species that citizen scientists can confidently identify in the field. They don't require microscopy or DNA analysis to know what they are. And they can also be verified by photos. And many other fungi that we come across either cannot be identified visually, or they haven't yet been described in science. So after the time surveys are complete, citizen scientists record the target species found in each transect to specific projects that we've set up on iNaturalist, as well as on a paper survey sheet. And we simultaneously have members of the project team undertaking surveys of the substrate, canopy and habitat, so that we can correlate the fungi found with the bushland condition. And we'll also be trialling whether the methodology works in the drier areas of the Murraylands and Riverland, and developing an education module with the Murraylands Riverland education team. In terms of recruiting volunteers, our strategy has been to partner with bush care organisations at different sites. So at our Trees for Life sites, we're working with Bush for Life volunteers and staff. At another site, we've been working with Hills Biodiversity and their volunteers. And other sites at national parks and conservation parks will be working very closely with the friends groups there. And the benefit of working with existing site volunteers is that they already have a connection with the site, and learning about fungi at the site really deepens their understanding of the ecosystem that they're working to restore and protect. And it also means that they're more likely to stay engaged in the project with more of a sense of ownership and an incentive to see the differences in fungi over time. As we need 10 to 12 people for each survey, we've supplemented numbers with members, interested members of the Adelaide Fungal Studies group and other members of the general public through promotion in the Fungi Map e-news, local environment centres and landscape boards. We've had plenty of challenges in our first year, especially in the technical challenges of surveying fungi, but some of the citizen science challenges we've encountered are sort of needing to pitch training in different ways for different learning styles. So this year we held an initial training session for everyone, supplemented by a written training pack, and then some training by staff on the day. But in future we definitely need to offer a video for people who are more visual learners, as well as continuing with the written information and the sort of practical demonstrations on site. We found that not everyone has the same sense of the importance of following the methodology exactly, and we sort of need to keep, reminding people of that or matching them up with someone that's done it before. Also, people have different abilities, and some people do tend to be a bit more meticulous than other people when it comes to recording, some people tend to be less meticulous when it comes to recording. So we've sort of had to develop some extra layers of data checks to make sure the data is robust, but this means that a little bit more staff input has been required. In terms of the technology, whilst we're using the iNaturalist platform, which is great, people are recording on their personal phones, and there's just amazing variations in things like connectivity, like at a site. Some people can connect to our projects, some people can't, which means some people can complete everything then and there. Other people have to finish their recording later at home, and it might happen, days or weeks later. There's all this kind of variation. Also variation in people's familiarity with iNaturalist, and we've sort of counteracted that by pairing people up with pairing them up with someone who is more confident with iNaturalist. And we've also found it tends to be quite a long survey day when we're surveying a couple of different areas, and the recording to iNaturalist can get a little bit repetitive for some people, and they can just sort of lose concentration after a while. We've tried to counteract that by looking at ways to reduce repetition and make the day more varied. At one of our later surveys, we had a much larger group, so we're able to split the group and have half the group doing the time surveys, and the other half just wandering around looking at fungi and chatting about fungi with experts. And that seemed to work really well, just to have that focused time on the surveys and then the less focused time, and sort of a few more breaks and opportunities to chat as well. So we're pleased with our progress this year, in particular the enthusiasm shown by citizen scientists for the project, and the fact that we've now educated and significantly upskilled these citizen scientists to be able to identify both the 25 target species as well as quite a few other species that we've found on the day. The project has brought together First Nations partners and mycologists and others with an interest in fungi. It's the first time citizen scientists have been looking for lichens as part of fungi research, as far as we're aware. And this year's 3 trial sites have helped us work out what our site requirements are for the remaining 7 sites. The main challenge this year has been many of our target species being absent or not as prevalent as we would have expected at these sites, and it's unclear if this is due to the fact that we had an extremely dry year or whether they're actually absent from the sites. And we had a risk management strategy of piloting at just 3 sites in the first year, and we're glad we did that because we needed to. So we haven't yet finished analysing the data from this first pilot year, but this will inform changes to the methodology and the target species for years 2 and 3. We look forward to continuing to share our citizen science journey with you all over the next few years. Thanks very much.